morning all. Let me continue the study of amino acids and proteins. As you know, amino acids are small molecules, proteins are big molecules. Amino acids make protein molecules by chemically combining with each other in large number. Thousands of amino acids can combine chemically to form a macromolecule. Let us see that this one protein molecule is a linear molecule. These are, say, amino acids. They chemically combine in a linear fashion. A linear molecule is a big linear macromolecule is obtained. And these protein molecules, a macromolecule, in nature, this is a formation from the amino acids. In nature, that is in all living beings, we have seen a wide variety of protein molecules. Not one protein molecule, a wide variety of protein molecules, different billions of protein molecules. The different protein molecules, it's all because of the sequence of amino acid. What is sequence? What amino acid? Next, what is amino acid? What is the next amino acid? There are 20 amino acids, as I said. Only 20 amino acids make all proteins. These 20 amino acids, in what sequence they arrange? That is repetition, of course, amino acids. In what fashion? In what fashion? In what sequence or what fashion the amino acids are arranged in the molecular, that is in linear fashion. That is a sequence important. The sequence will determine what is a protein molecule. That is one factor. And the other factor is the molecular size, protein molecule size. It depends on how many amino acids present in a single molecule. Whether the amino acids present in hundreds or thousands. Small molecules are there, big molecules are also there. So on that basis also we have get different protein molecules. That wide variety of protein molecules is all because of, that is, different protein molecules. We have seen protein molecules in all living beings, in all animals, in all plants, and also in microbes. These protein molecules, wide variety of protein molecules. It's all because of the sequence of amino acids and the size of protein molecule. And these protein molecules have what is that, a special character, is a God gift, is a nature gift. These protein molecules have properties, a fashionate property. These protein molecules exhibit what is that, a wide variety of biological functions. Different molecules have their, what is that, they have different molecular structure. A macromolecule is folding into form a different molecular structure. And these different molecular structures are different biological functions. They have wide variety of biological functions in the living system. This wide variety of biological function, that is, the activity of all the organism, whether it is activity of animal or activity of plant or activity of microbe, they have their own activity, daily activity, their life activity. It is all because of a resultant of all the biological functions of these proteins, a special characters, molecular characters. A resultant, a combined effect of the biological functions of all the proteins in the system will have what is that is seen in the form of the activities of the animals, activities of the plant, and activities of the microbes. Whether it may be an activity of the survival mode, or what is that, it may be a daily life survival, and any other activities of life. It may be a survival of microbe survival by infection other animals. This is all because of the protein molecules which favor them. We have their own protein molecules. Those protein molecules support us for our daily activities, for our survival. And plants have their own set of protein molecules. They favor what is that in support of plant existence, the survival. And microbes, whether virus, bacteria, fungus, parasite, what is that? They have the set of what is that protein molecules. Those molecules will help them to survive in the nature. That is the characterizing what is that of the protein molecules. There is wide variety of protein molecules. And these protein molecules were actually synthesized in the biological system. It is in this cell. It is a cell I written now. This is a cell like this animal cell. Example, plants are also there. Where is the site of the synthesis of protein molecule? Inside this cell. They have seen that the cell contains what is that compartments, different compartments, cellular compartments. So they explain the functions of the cellular compartments or organelles. They explain their different organelles they have their own specific function. They have what is that? We have nucleus. We have endoplasmic reticulum. There are two types, rough and smooth. Lysosomal bodies, cytoplasm is there, fluid mechanism contains so many nutrients and water. And here mitochondria, they have their own function. Golgi bodies, they have their own function. And like that, cell membrane, they have own functions. And every organ, every compartment has a membrane bound. This is a membrane like cell membrane. This nucleus has a membrane, this cell membrane like cell membrane. They have membranes. And they have their own, what is that? But they have their own functions. And this cell is what is that? Well organized. It is all because of highly coordinated function. Highly coordinated, what is that? A kind of a coordination among all the compartments, among all the organelles. Highly coordinated. And this coordination function together put into a cellular activity, entire cellular activity. It is under the control of course, central nervous system, the brain. Every cell, all cells in the organ are well controlled under the central nervous system. That is receiving signal and sending signal whenever and wherever. What is that? That is cell has that kind of what is that under control through signals, through neurons. That is electrical signal. That is the neuronal signals. I say that sensory signals. And this is what I mean talking about the compartments, different compartments in the polymer. Now I want to see that which compartment we have to concentrate. This compartment, these organelles. These organelles contains what is that? Endoplasmic reticulum. They are called as rough endoplasmic reticulum. Here endoplasmic reticulum is attached with so many ribosomal bodies. Let us tell you what is ribosomes afterwards. And also ribosomes are here attached to this organ as well as, and free ribosomes are also there in the cytoplasm. That is in fluid milk all over the ribosomes, ribosomal bodies in the Golgi, that is endoplasmic reticulum. On this ribosome, on these bodies, on these organelles, the protein synthesis is taking place. Let me explain the mechanism of protein synthesis on this ribosome, at this ribosomes later. On what basis the protein molecules are made in the cell at this site? That is, it is not as the cell wish, it is in accordance with the wish and will of the central nervous system. That is, central nervous system will decide what molecules of protein the cell is needed, or what molecules of protein the other cell, of course, the other cells of the body needed. On that basis, what is that? The central nervous system will give a signal. First is sense, that is sensory organs. So, sense what is deficient. These are cell metaphors. What is protein molecules, of course. What is deficient and what is needed for the cell? It is first sense the central nervous system accordingly gives a signal to a cell. That is signal through what is that? The tissue. That is tissue and then to cell. Through membrane and via this media. This contains all signal molecules are there. They have their only signal. It is called up to this is a neuronal signal, and from then onwards, what is that? They have their own molecular signals. The molecules. Signaling the nucleus. Nucleus contains what? What is the purpose of nucleus? This nucleus, no. Of course, placenta is dense material is called nucleolus, and this nucleus and also contains nucleoplasm, a fluid material like the cytoplasm. They are fluid material containing all the nutrients for the activity and all the enzymes and proteins what is needed for this activity inside the nucleus. Just like what is this? A fluid containing so many nutrients. What are the nutrients? Of course, we will discuss it later. And what is there in the nucleus? In the nucleus, very very important, the genetic material is stored up. The DNA molecules are stored up. Genetic information is stored up safely. Every cell, if you take a cell of any organ, all the cells of the organs contains what is that? 23 pairs of chromosomes or 46 individual chromosomal bodies. 46 chromosomes are there. 23 all cells of the human body, whether it is a bone cell or what is that skin, muscle, heart, liver, tissue, any cell or what is that? If you think about is that it is all cell contains 46 chromosomes and 23 has come from mother, 23 has come father, this combination. So this 46 chromosomes present in all the cells, but the genetic what is genetic information? A genetic language, some kind of language is stored in that in the form of molecules, macromolecules. Information, that is information, just like computer, no, the hardware information, that is material is non-living things. So just like the information, what is that? That is biological, no? How the biological, what is that? The biological information of the organism is stored in this material. It contains, this nucleus contains 46 chromosomes, 46 DNA molecules, 46 DNA macromolecules are there. And in this DNA molecules, a big molecule, polymer molecule, a macromolecule is even bigger than what is a protein molecules, very big protein molecule, each big molecule. And the information or that language, biological language or the information is stored in the big molecule. That is called genetic information, called genetic material. The chromosomes are genetic materials. So here the materials are there. Accordingly, what is our central nervous system will give the instruction to genetic material what gene and what chromosome supposed to work in that cell. That is decided by central nervous system. And what gene to be opened, because all genes are not open in all cells. In organ, only particular genes are open to make what is that particular proteins, because different cells of the organs will make different proteins, of course. So accordingly, what is that? Only that gene is open. Accordingly, that gene will make protein molecules. Let us go in detail how the protein molecules produce in accordance with the information given to this genetic material. What gene to be opened and accordingly what protein is synthesized. That information. Now understand, very important, that is need to be understand. What is the genetic information or the biological information that is in the DNA molecule? Let us understand that language or information, how it is stored, how the genetic information stored in the big molecule, that is a DNA molecule. What is just a brief let us go a little brief into the DNA molecule. What is DNA molecule? DNA molecule is a big polymer molecules, macromolecule. It is obtained by polymerization of nucleotides. Just like polymerization of amino acids, it became macromolecule. Here, monomer is amino acid. Monomers are amino acid and polymer is protein. In DNA, monomer is a nucleotide. This is a nucleotide. Polymer is DNA. What is monomer? Let us see that this is a monomer like this. Amino acids, there is different structure like 20 amino acids that 20 amino acid molecular structure I explained already. Now this is what is that a nucleotide. Molecular structure of nucleotide. It contains three components again. Three components, three simple components, chemical components combined to form one nucleotide. What is that? Three components. This is one component. Is a sugar. Sugar means pentose sugar. Pentose means containing 5 carbons. Carbon is here. Carbon, 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 carbon. 5 carbon atoms. And in cyclic, molecular structure is cyclic structure. This is a ring structure. Molecular ring. And it contains what is that? Oxygen is there. Ringing through oxygen. This is called pentose sugar. 5-membered ring. Deoxyribose. This OH is there. And OH is not there in place of O, is removed from there. Otherwise it becomes ribose. This ribose is also important in RNA. This is deoxyribose present in DNA molecule. This is ribose, deoxyribose. This component is called as sugar or deoxyribose. This combined with one more chemical component is called base. This base are written as B. So there are base, not one base, there are 3 bases, there are 4 bases are there. This is one base. Let me go in details afterwards. This is one component, another component. And one more component, simple component is phosphoric acid or phosphate group. POOOH, phosphoric acid. This combined chemically. The OH is there and this OH is there. Combined chemically with the elimination part of these two combine. Phosphoric acid and that is sugar, pentose sugar combined chemically. And this sugar chemically combined with base, forming chemical bond with base here. So it is a combination of 3 simple components, base, sugar and phosphate, form a nucleotide. That is what? Monomer. And what is this base? It may be this or this or this, all possible in DNA. This base, look at that. They are organic molecules containing nitrogen, of course. That is just like a benzene ring, not a benzene. Just like benzene ring, we have not N here, CH2. If you don't write this, CH2 at the corner, CH double because of double bond, CH3, C double O. Bond into that is ketone like NH. This molecule contains 2 nitrogen. Any organic molecule containing nitrogen is called as a base. Any organic molecule which contains a nitrogen atom, it is called as, because basic activity, that is produces a molecule, a basic character in it. Base, it is called as thymine. And one more molecular structure layer, just like benzene ring like this, not benzene. There is one more ring. It is penta, that is 5-member ring. This is 6-member ring. 6 bonds in cycle, 5 bonds in cycle, 5-member. Here NH, CH, here CH is there, N is here, CH2, N, CH, N. That is all contains 4 nitrogen. It is called adenine. The base is called as adenine. And one more, look at this, the structure like this, cytosine, nitrogen, this is H, CH, CO. One more base, guanine. Look at that, benzene like, and that is the ring of what is that? 5-member ring, 6-member ring, containing nitrogen, 5 nitrogen are there. This is purine base, these two. This is called pyrimidine bases. It is called cytosine guanine. This is a molecular structure of these bases. Nucleotide means there are 4 possible nucleotides with this base, one nucleotide with other base. This is common structure. With different bases, we have different nucleotides. We have 4 different nucleotides. Here, 20 different protein molecules, 20 different amino acids, but here 4 different bases, 4 different nucleotides. Chemically combined, polymerization. to produce a big molecule. The polymerization, what is the polymerization? The reaction taking place is a nucleotide reacting of the nucleotide at the two ends. This one end is a reactive, other end is a reactive. This is alcoholic group. Alcoholic group, this is hydroxyl group of the phosphate. Here like this. And reaction between the hydroxyl group of one nucleotide and the OH group of this nucleotide, other nucleotide here, one more nucleotide like this, like that. This is one nucleotide, another nucleotide. Chemical reaction through between this group and this group. This group OH is there, OH is there, and this OH and OH, they chemically react with the elimination of water, and a new chemical bond arises. This chemical bond will give a binding between these nucleotide. This nucleotide is binding with this nucleotide. Through this chemical bond. This chemical bond in this nucleotide is called as diester. Poly, what is that? Phosphate diester through phosphate. It is known as phosphate diester. It is through phosphate. In previous case, the bond between amino acids is called as peptide bond. That is the bond I showed that. This bond is called as phosphate diester. This is linking two nucleotides. Likewise, the partner is a nucleotides. What is that? Keep on, what is that? Adding with the reaction, adding this side of the reaction, other nucleotide, they grow in size, linear molecule. It is a big linear molecules of time. And molecule, what is that? Base. These bases, what is that? 4 different bases are possible. Now you can see a big polymer molecule, linear molecule, a macromolecule, linear molecule is obtained. Let us call this as a linear molecule. I have not shown this structure, this chemical structure. There is a line like this containing what is that? Sugar, phosphate, and base I mentioned here. A, G, T, C. This is one nucleotide with A. This is another nucleotide with base G. Another nucleotide with base T. What is one? Bases actually, because they are important bases. That one is not important. This is one big linear molecule, a giant molecule. And imagine how many nucleotides combine to form that molecule. Not thousands or lakhs, here millions, 50 millions or 100 millions of nucleotides. There is a billions of nucleotides combined to form a big molecule. What is that? Big macromolecule. A DNA, what is that? A DNA strand like this. After this DNA strand is obtained, that is the linear molecule is obtained. This linear molecule usually will not exist in a single strand. There is one more DNA molecule like this, one more polynucleotide. It's a polynucleotide, one more polynucleotide with these bases common. That is sequence of bases. These are called sequence of bases in this macromolecule, polynucleotide. And there are always what is a greater attraction. There is always an association between this linear and this linear molecule. What is that? This polynucleotide and this polynucleotide. It is all because of some kind of a weak chemical interaction between them. Which one arises from which? It is between the bases of this and bases of this. That is adenine and thymine. There is always there is a possibility of formation of 3. What is that chemical bond? These chemical bonds are called hydrogen bonds, intermolecular hydrogen bonds through hydrogen, because we have hydrogens and oxygen, we have hydrogens and nitrogen. This hydrogen can interact with the oxygen, this hydrogen can interact with the nitrogen of this other base. That interaction, that is an interaction, chemical bond. It is not a complete chemical bond, it is a partial chemical bond. Bonding through hydrogen, this is called hydrogen bond. So this can form three hydrogen bonds, intermolecular hydrogen bond. This intermolecular hydrogen bond, what is that? What is the binding between these two? Binds these bases, and these bases binding through two hydrogen bonds. Therefore, two dotted lines. TA or AT, three bonds. GC or GC. Because these are the binding between them. But A and AC cannot bind. A and T. Like this, we have polynucleotide, individual polynucleotide, again combined like a ladder. Two, what is that? Nucleotide molecules combined or associated just like a ladder. And this ladder will not be linear, will be twisted into what is that? A double helical structure. That is, twisting is a definite order is there. There is always a definite order in the twisting of molecule. This is called double helix. Their DNA molecule, the structure of DNA molecule is a double helix structure like this. Just like a ladder, twisted ladder in a definite pattern. This is called DNA molecule. Contains two strands, individual two polypeptide molecules. There are series of bases like this, sequence of bases. On each DNA molecule, each chromosome, actual structure, when you remove this is the structure of the DNA molecule. How many chromosomes are there? 46 chromosomes are there. 46 DNA molecule like this. They are different. 46 DNA molecules are different because of different sequences. The sequence, just like protein molecule, they are order of arrangement, the bases here. That are different sequence and different size. That is smaller size and bigger size. So among the 46 chromosomes, here the smallest possible chromosome, it contains 100 million nucleotides, nearly 1 billion nucleotides, smallest possible chromosomes. And the largest possible chromosome, which contains 500 million nucleotides. Okay, 500 million nucleotides, a big molecule. There is difference. 100 millions, that is 200 millions, 300 millions, 500, there are different sizes we have. So among the 46 chromosomes, all chromosomes are DNA molecularly. All the DNA molecules are produced by the nucleotide, four different nucleotides, 4 different bases, only 4 nucleotides combined in sequence. They differentiate, that is chromosomal bodies, that is chromosome molecules are differentiated on the basis of the sequence of the bases in the double strand and the size of the DNA molecule. Size means it depends on how many nucleotides combine to form a macromolecule like this. There are 100, 200 million or 500 million like that. So that is 46 chromosomes are there. That is the structure of DNA molecule. Now I want to understand what is the going back because we are going into the understanding of genetic language or biological language. What is meant by genetic information? That is called as a series of base. A sequence of base is actually a genetic language. We are talking in terms of the other way because this is the way it is not written because the sequence, that is a linear sequence of bases. We talk in terms of what is that? This sequence. Not in single sequence, in terms of what is that? 3 nucleotides, 3 bases like this. This is always in series, individual, but with the 3 like this. Why 3 like this? Very important. These 3 together with just like A, T, T, that is one sequence. That is in this order. Adenine, thymine, that is in this sequence. So that is called as one codon. I'll tell you what is meant by codon. It is called as another codon. It is another codon. It is another codon. A sequence of codons there is. sequence of this and this and this called sequence. In this sequence we talk about. What is codon? Very important, because this codon, what is that? This one codon, this, that is 3 amino acids together, can combine with one single amino acid. That's what is that? Codon. What is codon? Contains 3 bases in this sequence. It's called triplet. It's also called as triplet. These, what is that? This bases, no, together, this can combine with a single, what is that? One single amino acid. What is the basis combine? Just like what is that? Intermolecular hydrogen bonding. So this can have, what is that? Produces intermolecular hydrogen bonding with what is amino acid, individual amino acid. An amino acid can, only one amino, not two. One amino acid can bind at this. One amino acid can bind at this. Therefore, you talk in terms of triplet, not single. Single cannot bind. 3 together can bind with one amino acid. It is called as one codon, one long acid. That is just like ABC, one codon. And of course, in terms of RNA, of course. Later, this is DNA. How many bases are there? 4 different bases are there. Using 4 different, using 4 bases, there are series of them. That is millions, not a series in millions are there. How many triplets the 4 bases can make? This is what is a permutation and combination. 64 codons, because 4 bases. This order, no, this because this order is different. This order, this is like this. This order, no. The order of arrangement of this, no, 3 at a time. Among 4 bases, 3 at a time. The order. How many bodies are? 4 bases are there, 3 at a time. 4 to the 4 of 64 different codons are possible. Any DNA molecule is like the 64 codons are there. What is a codon like this? This codon is same is wherever it's called as its own codon. One codon. 64 codons are there, and these 64 codons can, what is that? Can read 20 amino, because 64 codons are there in the genetic material. 64 codons are there in genetic material. That is called genetic information. And always talk about what is that? A sequence of codons. Not sequence of bases. Sequence of codons. This sequence of codons will decide what is the sequence of amino acid molecule. It will about amino acid get how the amino acid will get there, protein molecule will get by combination of amino acid later. That is what we see, that this is a sequence of codon. This is 45. Among the 64, 3 usually will not involve in the synthesis of protein. They are called as stop codons. These codons will not be usage in the synthesis of what is the protein molecule. They stop after the, there is wait, because after when the molecule begins to synthesize, where to stop? It requires some break. The break requires these codons are there. That is called a stop. So only 61 codons can read 20 amino acids. Here 61 codons can combine with 20 amino acids. Here also specificity is there. 61 cannot combine with one amino acid. One amino acid cannot combine with all the 61. That is also what is specificity is there. What is specificity? Example, if you take lysine, this amino acid, this can combine, this can bind with either this codon or this codon. There are 61 codons are there, but cannot bind with any codon. It can bind with either this codon or this codon. This is what is that? A series of thymines. That is adenine, guanine. Specific here also. That is, of course, one amino acid can more than, because 61 codon is a 20 here. One amino acid can take more than one codon to read. And example, alanine is a amino acid. So this alanine, what is that, can bind with, there are 4 possibilities are there in this amino acid, not one. That is one codon, GCU, GCC, GCC. 4. So that by some amino acid only 3 codons. So like by 2 or 3 or 4, these 20 amino acids are specific with certain codes. The codes may be 2 codes, 2 codons, or 3 codons, or 4 codons. So this is what specification of amino acid, each one. And this is about the DNA sequence I'm writing. This is language. What is Aisha? I want the information. What is the information? The information, or what is that? The molecular language is, this is the molecular language. A big DNA molecule is there. The sequence of this triplet, not the basic sequence of this triplet. There is a definite sequence is there. This sequence of difference in thousands, about 1000 or 2000 sequence is called one gene. That is language. One gene is thousands of these codons in one sequence. It is called as a gene. That gene can make one protein molecule like this. There is a language. It is called molecular language. So it is called as molecular language. So that molecular language is hidden in the nucleus. That is genetic language, genetic material, genetic information hidden in the nucleus. It is opened whenever it is required. It is opened by sensing through what is that? By the central nervous system. That gene is open, not all the genes. Only that gene is open. That gene will make a protein molecule. Come out, that gene, that copy will come out and make a protein molecule on this cytosol body. The protein molecule will come out. So you see that how many protein molecules is required, very important. What protein molecule is required? How many is required? There is also regulation is there. Everything. The own regulation control by the nervous system. When to stop, when to start, when to stop. Whenever the required amount of protein molecules are produced, it will come, it will automatically stop. That is very important. That is called genetic language, genetic information. It is called as central dogma of the life, central dogma of the molecular biophysics. Language, what is stored inside. Information is stored inside. All the information is stored inside, but only some information will act to make what is a protein molecules. What protein molecules is required? It is called as central dogma of life. That is in all living things it happens. It is all in plants, it is all in animals, all animals, and also in some microbes. So this is what is happening here. So now DNA molecule is here. This is a DNA molecule with the genetic information. What is a gene or a gene? Is what is one gene? One gene is what is that? A sequence of these triplets in thousands. There is 100 or 1000, depends on what molecule is produced. Hundreds or 2000. That sequence, it is called as a gene, is a language. Different genes are there. Different genes are there. Different languages. And accordingly, a gene can make a protein molecule. Another gene can make another protein molecule. Another protein. Different genes make different molecules. And what is the process of making molecules, protein molecules? Not this DNA will come out and do the molecules. Well, another mechanism is involved. This DNA, what is that? Is under what is that? DNA molecule will remain in the what is that? In the nucleus. It will not come out. Whenever the information is received to the central nervous system to make that particular protein, DNA will do what is that? The DNA, what is that? Will open that gene. How will open? It is requires what is that? Separation of these strands. Where that gene is stored. Which chromosome? Which chromosome to be involved? And what gene to be opened? Only that gene is opened. This will what is that separation? What is that? Is a replication. That is DNA molecules what is that separated. It is opposed by the action of enzyme because every what is that? For every action there is enzyme because this nucleoplasm contains all plausible enzymes are there. All protein molecules are there. All nutrients are there. What nutrients? It all contains all nucleotides. These nucleotides. To make what is that? DNA molecule as well as RNA molecule. It all nucleotides. And here nucleotides either it can be synthesized by the same organism. It can be synthesized by the liver. In human being, the nucleotides are synthesized in the liver and go to blood and then distributed to all the cells. That is one way. And the plant can also prepare nucleotides in the plant. That is a chloroplast. I told you nucleotides were prepare or synthesized in the chloroplast. It can be taken as a source through food, either through as a source or synthesized by itself in the liver. Nucleotides are there. Nucleotides transfer from what is that blood to tissue, tissue into the cytoplasm, and via into migrate into the nucleoplasm. This is concentrated. All nucleotides are concentrated here, and all protein molecules or any nutrients or water, everything is stored up here for the activity of DNA molecule. So this DNA, whenever receive the signal and what gene to be open, it is opened here, separated. That is called as DNA binding, DNA replication, that is. And this, after this opening, it will make a copy of this RNA, just like similar to this molecule. The molecule is called, why it is called RNA, same nucleotide molecules. If once it is opened, is separated, and these are left free, this can combine with a nucleotide. What nucleotide? RNA nucleotide. What is RNA nucleotide? DNA nucleotide is, it is a nucleotide with this base and this base and this base and this base. What is RNA nucleotide? Only one difference. In RNA, base is, except this, these 4 bases, again 4 bases. This base in RNA uses this or this or this or this. 4 bases are this. But T is replaced by uracil. There is only difference. In RNA, we don't see this T. In place of T, uracil come. 4 different nucleotides. In RNA, one more difference here. Here CH2 is there, CHOH is there. Ribose sugar. What is RNA? Ribonucleic acid. What is DNA? Deoxynucleic acid. They call nucleic acid. Molecules called as nucleic acid. Ribose sugar. Deox means deoxyribose sugar. The difference is here on place. In RNA molecule, just like same polynucleotide. Once it is open, and one more enzyme, it is there already in the called RNA polymerase. It will make RNA. It polymerizes RNA using these nucleotides. And it is polymerization. Here taken cytoplasm contains what is nucleotide. Here combined with this nucleotide form a chemical bond. They form a combine because combination alignment. Nucleotide shift with the alignment oriented for a chemical reaction in that direction. That is the proper orientation for a chemical reaction. So here a nucleotide with a complementary T can A sit here, and C with G and A with U like this nucleotide. So now nucleotide can combine. With combination oriented reaction between them, and you get a polymer like this. Not, it is A, U, G, C, G, U like this. G, C, A, T. In place of T, U. That is a polynucleotide. It is called RNA molecule. RNA molecule not as big as the DNA molecule. It contains about thousands of nucleotides. That is called mRNA. That is called mRNA. What is mRNA? After this making mRNA, once open G, that is open, and after making RNA, that is particular gene is read. The particular gene is read in the form of mRNA. It is called transcription. Transcribed. This language gene is translated into this gene. It is called RNA because DNA cannot come out. It remain the same. It will send a copy of this DNA in the form of RNA. This RNA molecule can come out through the pores of nuclear membrane. Nuclear membrane is always highly porous. So through which protein molecules enter into and RNA molecules come out like this. There is an exchange is there between the nucleoplasm of the nucleus and the cytoplasm of the cell. The RNA molecules after synthesis come out. This RNA is same as this is what is the language. The same language. In place of T it is U. That is what language is there. That is a series of what is that? This genetic, this gene is a series of codons. How many codons? Maybe hundreds of thousands. Depends on which size of protein molecule, what sequence of protein molecule is prepared. This come out as mRNA. It is called messenger RNA. Once the messenger RNA has come out of the nucleus after preparation, this is again closed. After replication, what is that? Again one more enzyme. What is that? Will bind these into back into what is the same structure. Remains safe and stable. So whenever it is take the signal or sense, what is that? It will open. Only a particular gene is opened, and it is that language is read and translated into another molecule is called mRNA. And again it's what is that? Go back to the same structure and remains what is that? These molecules actually, before what is the binding us, this actually stabilizes these molecules, DNA molecules are not like this a linear molecule. This is what is twisted around protein balls. Contains molecules of proteins here. They are called stones. To make that molecular stability, to maintain stability, this is always twisted like this. That is twisting around a ball, protein ball, and remains at a small space. All chromosomes are occupied in a small space. Whenever it takes a signal, it rewinds and becomes a linear molecule, and it is again separated. That is replication, and send that language in the form of another molecule. Copy, and this copy again will float back. That is another enzyme is read to what is that again make is that is same structure, and this polymerization takes place in presence of other enzyme, RNA polymerase. Once this molecule is come out, what is it come out? Is sit on that what is that site of ribosomes. That is what the language. It is transcribed. What is transcription? That language as language in the DNA molecule. Yes, transcripted. What is that? Transcripted into the other copy as RNA, and this can come out. RNA can come out. Small molecule. And once the RNA has come out into the what is that cytoplasm, what is that? These ribosomal bodies are already is in here by now. These ribosomal bodies, what is attack? Their affinity towards. Why ribosomal bodies? These ribosomal bodies are of course protein bodies. They contains an assembly of protein molecules. There are two protein bodies are there. This is one large subunit and small subunit. It contains what is that? Ribosomal RNA. It contains some RNA. RNA is a RNA. It combines with RNA, some RNA called ribosomal RNA. That ribosomal RNA, some code is there. That is one or two codes are there. That ribosomal RNA can, but because of presence of RNA and RNA, here RNA is there, they can bind with another RNA. This is a linear, what is that language, already transcribed and come out for the making protein, particular protein. So once it comes out, these two bodies combine like this. After this combine, what is associated? At the initiation, very important, at the point of initiation, at the point of synthesis, begin there, at the point of synthesis. And once they bind, then this, what is that? That is machinery, what is that? That's like a cellular machinery begins to form. What is that? What is the cellular mechanism? That's the same, it is same as this mechanism of synthesis. What is I'm showing that now? That's I will explain later. This mechanism is similar to what is that? The mechanism what you see in the industrial area, industrial field. Nowadays all industrial machines, that is industry with all components around us are computer automated. That is automated machines are there. In such a way that they take the raw material by themselves and process at different levels, different parts, all of them are automated. Specific, that is each part is specific function, is computer automated. Finally, the product, the desired product will come out with all this processing at different units. It is automated. You might have seen directly in the industry, or you might have seen through what is that media, mass media. So that is every industry in the world, it is all industrial equipments or industrial machines and their parts are automated. Automatic work like that is same mechanism here working. Auto machinery here. What is auto machinery? These ribosomes are what is the once they combine with mRNA like this, and this is a language that is that codons, the gene, and combination with this, what is that? Here begin what one more RNA is there, that is called transfer RNA. What is transfer RNA? A RNA molecule folding and exposing only one codon outside. This codon, that is codon. These are no, that is this RNA molecule can bind with what is that? They are bind with one amino acid. This is a codon. Next page. This codon and this codon, what is that? They attract. That is only specific codon. These are codons on this mRNA. That is a single codon. Each tRNA is a single codon. It can carry amino acid, one amino acid. How many tRNAs are possible? Because there are either 61 codons, whether here or here. Therefore, there must be 61 tRNAs. Each RNA is only one codon. And how many codons? That is, there must be 61 tRNA. What is tRNA? A small molecule like this, with exposing only what is that? One codon outside to interact with any other codon in the mRNA. So this codon, this can carry one amino acid. A T can combine with one amino acid. These tRNAs are present in cytoplasm. All 61 tRNAs are present in cytoplasm. They are all carrying their own amino acids. 2 can carry one amino acid, or 3 can carry one same amino acid like this. Amino acids. They are there. Once they combine, what is the combination, what is the association like this? There is a provision called is that the molecule, that is tRNA molecules enter into this molecule. There is a provision to exit in this molecule. In what fashion they exit? Accordingly, see that there are codon is there, codon is there, codon is there. If this codon suits with this codon to combine, not all codons can suit with this. Codon and anticodon, it is complementary basis. And this base is complementary to this codon can combine with this codon. That's why this will collect this tRNA. This tRNA come inside and sit binding with this, with the amino acid over there. Right? Like this already, previous one it is here shifting. And this association or what is that, the molecular binding is not stationary, it will keep moving. These two bodies are keep moving over the mRNA, and these things are moving inside like this in this direction. While moving, what is that, when they sit here, already sit here, moving now, this sit here, when these amino acids, what is that, these two amino acids come closer in a proper orientation to chemically connect. So this is a tRNA correspond to this sequence, this sequence, this sequence. And when the chemical reaction takes place, and these two bind and move like this molecule. And as these molecules, after this is loaded, this is unloaded. After the amino acid, what is that? It will lose the amino acid, will come like this. It will go here as a, what is that, into the form of protein molecule. It will, after unloading, it is come out like this, like this. So while it is keep moving, this is unloaded will come out, loaded will go inside. Loaded in accordance with the sequence, not as any tRNA can come here. Only amino acid of this sequence can combine here. Only amino acid, particular amino acid come here. So this is like what is the protein molecule synthesis. This is the last subunit. And likewise, a protein molecule with what is that? The sequence of amino acid. This sequence of amino acid is directly related to the sequence of the codes, codons or genetic code. And this sequence indirectly, what is that? This genetic code in this case. So this genetic code as read in the form of this protein molecule. This is a protein molecule of what is that? Once a protein molecule of one particular specific protein molecule is synthesized with this sequence of molecules. And how many amino acid molecules can combine? It depends on the size of the mRNA. How many codons are there? Hundreds are there, thousands are there. And after when it comes to the end of stop, that is stop, it is done by these codons. When you arise at the end of either this codon or this codon, it will stop synthesizing. And after the stop, what is separated. These individual what is smaller will go separated, and this mRNA molecule is left out. It will find out another tRNA like this, what is mRNA like this. These ribosomes will go back, it is separated and go back. And after synthesis, what is that? A protein molecule is obtained. It is called central dogma of life. Central dogma of molecular biophysics mechanism. So this is how a protein molecule of one specific sequence, a protein molecule of one particular size is obtained on the basis of the genetic information stored in the DNA molecule. The form factor. So this is what I am talking about, the central dogma of this. And this tRNA molecules, what is that? Where they present this tRNA and ribosomal bodies and ribosomal RNA, they are all synthesized in the nucleolus. This nucleolus make all ribosomes. After the ribosomes will come out and sit here. And also tRNAs, mRNAs, all synthesized by the DNA molecules. That is, the big large molecule is there. So synthesize and make what is that. So this is the synthesis of all RNA molecules, mainly mRNA, for synthesis of one particular molecules. It is called a central dogma. So this is how a protein molecule is synthesized in the cell, biological cell, which may be a plant cell or animal cell by microbe. There is always some mechanism is there is always some control is there is always some language stored there by DNA molecule. The language or genetic information is stored in the nucleus. It is all based on this information and based on the instruction given by the central nervous system. What molecules of protein to be synthesized. That is why the subject is synthesis of amino acids. Let us see in the next class. What are these molecules? What is that? I said wide variety of molecules, wide variety of functions are there. What are these functions? How they help the different animals in different way for their survival and their activities, daily activities. So let us see that in also macro, that is in macro organisms and microorganisms like virus. They exist now. The survival of virus, there is different. I think new virus is COVID virus. What is the survival mode of the COVID? Let us see in the next classes what is the function of these protein molecules in different organisms, whether microbes or macrobes. Thank you very much.